Delighted to welcome Professor Bonnie Bassler to deliver our first prize lecture of this conference. The prize medal is awarded to an outstanding microbiologist who is a global leader in their field and whose work has had a far-reaching impact beyond the discipline of microbiology. I can safely say in this case we have an outstanding recipient in Bonnie, who has spent her career investigating how bacteria talk to each other and orchestrate collective behaviors. Bonnie is a Squibb Professor and Chair of the Department of Molecular Biology at Princeton University. Her career awards and honors are simply too numerous to list here in the time available, and she has had numerous positions in service of our community, including being the President of the American Society for Microbiology, Chair of the American Academy of Microbiology Board of Governors, and a member of the National Science Board at the request of President Barack Obama. Welcome to Bonnie, whose prize lecture is entitled Quorum Sensing Across Domains, from Viruses to Bacteria to Eukaryotes. While her lecture will be delivered remotely, please do stay in your seats afterwards, as we will be having a live Q&A session afterwards led by one of her nominators for the prize, Dr. Chloe James.
Well, hello, and thank you to the Microbiology Society for awarding me this wonderful prize. It's an honor, it was a surprise, and it's an absolute delight that my team's work is being celebrated this way. So my heartfelt thanks. And I thank everybody for the opportunity to give this seminar. And without further ado, let me just get going and tell you about some new work from my group. So today I want to tell you about Quorum Sensing Communication and how it spans kingdoms. So to get going, let me just start by saying that my lab, always what we're trying to understand is how do bacteria get any bang for their buck? They're so small and inconsequential individually, yet they do all these powerful, terrible things like cause disease and all these miraculous things like be our partners in our microbiome and give us all kinds of bioproducts that we need for life. So harmful or beneficial, bacteria have all this power and we wonder how they manage it. And so what we think the answer is that bacteria communicate with one another, they count their numbers, and they act in groups. And this process is called quorum sensing, and it's sort of shown on this first cartoon. So what we think is that bacteria have to understand times when they're alone from times when they're in groups, and they behave differently based on those two scenarios. And the way they do it is with quorum sensing. So this is supposed to be my bacterial cell. And so at low cell density, bacteria want to have the program of gene expression going that's good for acting as an individual. So they're carrying out some subset of the tasks that they're capable of doing. And among the things they do is that they make and release small molecules depicted as these red triangles that we call autoinducers. And those molecules diffuse away, the bacteria can't detect them, and that says act as an individual. But then as the bacteria grow and divide, since all of the cells in the environment are making a share of the autoinducer molecule, the extracellular concentration of the molecule increases in proportion to cell number. And when the molecule hits a particular threshold amount, the bacteria detect it, and they infer from that detection moment that they must have neighbors around. So in unison, all of the bacteria change their gene expression and their behavior together, and they begin to carry out collective or group behaviors. So in fact, the bacteria have no idea how many cells are around. They're measuring the concentration of this chemical as a proxy for cell number. And we now know that quorum sensing is the norm in the bacterial kingdom. There are thousands and thousands of examples of quorum sensing bacteria. And typically in every bacterium that's been studied, somewhere between 200 and 600 genes are controlled by these circuits. So it's a big deal when a bacterium changes from going in alone to acting as part of a group. And so now we know a lot about these, or we know some things about these molecules as well. What my group thinks is it takes at least 3 quorum sensing molecules for bacteria to have a proper conversation. So I'm going to show you molecules from Vibrios because marine Vibrios was where quorum sensing was first discovered. So I'm showing you these very particular molecules. And so even though the bacteria, cross the bacterial kingdom, they change the molecules, they tinker with what they are, how many they use, how they get detected. But the themes that I'm telling you about how quorum sensing work, that seems to transcend all quorum sensing communication conversations as far as we know. So what I mean by that is there's always a molecule. So in this case, this homoserine lactone, there's some molecule that one species and only one species appears to make. And so this molecule enables intra-species communication. This is how I count my siblings. Then there's a molecule we discovered, this fatty acid, that all Vibrios make, but nobody but Vibrios makes, as far as we can tell. So we think this is the molecule that says the genus. So the second molecule says, I'm your cousin. The first molecule says, I'm your twin. And then the third molecule that we discovered on the bottom here, this is broadly made across the bacterial domain, gram negatives, gram positives, and all bacteria make exactly the same molecule. So we think this molecule has no species information in it. It says other. So what we now think is that these molecules encode something about cell number, but also something about how closely or distantly I am related to my neighbor. And the bacteria track those pieces of information and they respond accordingly. So now, if I put that part together, what we think the computation that bacteria do is the following. The first thing we think they do is they scan the environment and they ask, am I alone or am I in a group? So they sort of scan the environment for quorum sensing molecules. And that starts to set the program of gene expression that the bacteria carry out. But then the more sophisticated computation that they do is that they measure the ratios of those molecules that I just showed you. So at high cell density, not only do they know there's a lot of cells around, they're actually asking, is it me and my kin or is it the enemy? So they can distinguish self from other with these molecules, and they tailor these programs of gene expression they turn on and off based on whether they're surrounded by friend or foe. Right, so now we get it. Quorum sensing is about knowing when you're in a group, but also knowing who your neighbor is and acting appropriately based on both of those pieces of information. All right, so that's the background. And today I want to tell you a new story. And I'm going to stick with Vibrios, but it's going to be about this global pathogen that you've probably heard of, Vibrio cholera. So Vibrio cholera is an endemic pathogen that causes disease. You might know that you get cholera disease by drinking contaminated food or water. And cholera has to have quorum sensing to be a pathogen because quorum sensing controls infection and dissemination. And so we discovered quorum sensing in cholera, and I'm showing you a sort of simplified version of the quorum sensing circuit, just with the parts that you need to know today. So cholera causes this acute disease. You get it from contaminated food, and cholera's strategy is sort of to come in, guns blazing. It's really infectious. It gets in, it makes a biofilm on the cell surface, it expresses its virulence genes, and then quorum sensing kicks in. And at high cell density, cholera turns those genes off, turns on an escape program, and it escapes by the gazillions to infect the next patient. So it's all about dissemination. And so the way that works is at low cell density, when cholera infects the host, there's no autoinducers there. And so under that condition, cholera makes a set of small RNAs that we call QRR for quorum regulatory RNAs. These are like microRNAs in eukaryotes. These small RNAs allow biofilm formation, so that's how cholera sits on the intestine, and then the expression of virulence genes and toxins. So very, very infectious on the way in. So then cholera is in the intestine, it's infectious, and then as it grows, these autoinducers that I told you about kick in. And as far as we can tell, cholera only uses 2 of those autoinducers that were on my last slide. So at high cell density, the autoinducers accumulate and they get bound by these transmembrane bound receptors. And when the autoinducers are bound by the receptors, it flips a switch and cholera stops producing these small RNAs. And under that condition, it shuts off biofilm formation, shuts off virulence factor expression, but quorum sensing turns on the escape genes and out cholera comes to infect the next patient. right? So quorum sensing is controlling this switch that allows the bacterium to infect and then to leave. And so when we got about this far in our studies, a fantastic postdoc named Kai Pappenfort joined the lab. And he was really intrigued by these small RNAs, these very eukaryotic looking microRNAs that sat at the heart of the circuit. And so what Kai wondered is whether there could be more to it than the small RNAs that we had originally found. So he wanted to do an experiment to look for more quorum sensing regulated small RNAs. And so he did something really easy. He just did an RNA-seq at low cell density and at high cell density, looking for small RNAs that had a pattern of regulation in which they were expressed under one case, but not the other. So he looked for all the small RNAs in cholera, and sure enough, he found hundreds of small RNAs that were both expressed at low cell density and high cell density. So the cholera genome is loaded with small RNAs. These probably have fascinating functions, but we didn't study them because they were on at both low and high cell density. Then he found the small RNAs that we had already discovered. So at low cell density, the QRR small RNAs were expressed, but at high cell density, they were reduced. And that had to be the case. We already knew that, so that was sort of a built-in control for this experiment. So then Kai got one more small RNA, and it had the opposite pattern. It was expressed at high cell density, but not low cell density. And so since that looked like a quorum sensing regulated pattern, Kai got interested in this small RNA, and he named it VQMR for Vibrio Quorum RNA. So he wanted to place that into the circuit. So here's the components of the circuit that you know. At high cell density, biofilms shut off, virulence shuts off, and escape turns on. This small RNA, VQMR, is expressed at high cell density. And so the next thing Kai wanted to know is, what does it regulate? And he did a bunch of experiments, and they're all published already, so I'm just going to tell you the answer. Kai showed that this new small RNA repressed a gene called mRNA called VPST, which encodes the major regulator of biofilm formation. So what that means is that at high cell density, when this small RNA is turned on, biofilms shut off. And that should track with exactly what I've told you. So now what we're starting to think is that these three circuits, the one we knew about, and these two that we knew about, and then this new small RNA, all feed in to shut these biofilm genes off, and he showed its virulence genes as well at high cell density so cholera could escape. And so once Kai got that far and he proved that that was correct, that sort of left us with another problem, which is, well, what's regulating VQMR? How does it turn on at high cell density? Something must turn it on. And so Kai did a genetic screen to look for an activator that would turn on the VQMR small RNA. So I'm going to show you the strategy. So the idea is this VQMR small RNA turns on at high cell density. And we would guess that there must be an activator, right? And then this shuts off biofilm formation. And biofilms are hard to study. So what Kai decided to do was he just made a fusion from the VQMR promoter to the LAC-Z gene. So when this circuit is running, the bacteria turn blue. So this is sort of like genetics 101. You know, people have used this trick many, many times. So then what Kai did, so the cholera are blue at high cell density because this promoter is on. And so then what he did is he just did a random transposon mutagenesis of cholera, hoping to knock out this putative activator. The idea being, when this circuit is broken, then the bacteria wouldn't turn blue because this gene is off, they turn white. And so he did that experiment and he collected up about 20,000 mutants. And sure enough, he got white colonies on the plates. And when he sequenced what was in all of his white colonies where the transposon was, he got exactly one gene out of this screen. He got a transcription factor. So he got a DNA binding protein that he showed binds right at the VQMR promoter to turn it on. So this was the circuit, and he named this protein VQMA for Vibrio Quorum A. So this is the transcription factor that turns on VQMR, and then VQMR turns off biofilm formation along with these other quorum sensing circuits that I've already told you about. Okay, so now Kai was cooking with gas, but the problem was, this just sort of gave us the next level problem, is what is regulating this new transcription factor? So all of this only happens at high cell density. And so what I can tell you is that VQMA is expressed constitutively It's always there in the cell. And so when we saw that, we thought, well, okay, so the protein itself isn't, its amounts aren't regulated, but maybe its activity is regulating. And so we had to guess that there was something regulating the VQMA activity. So it's only on or working at high cell density to make this circuit complete. And so, of course, in my lab, when we have a protein whose activity changes, at high cell density, we of course think, oh, well, those are autoinducers. Autoinducers accumulate at high cell density. They make proteins change their activity and they turn on and off genes. And so we thought, okay, well, this is our kind of thing. We already have these autoinducers. So you'll remember, we know the identities of these autoinducers. Here they are again to remind you. And what I haven't told you is we can make these synthetically. We have buckets of these autoinducers in the lab, and we also have mutants in cholera that don't make one, don't make the other, or don't make both. So we have all kinds of ways to test whether autoinducers are controlling Kai's new circuit. And so I'm just going to show you one experiment that Kai did to try to find out if the autoinducers were playing a role. So what you'll remember is that we're going to guess that autoinducers are going to turn on this VQMR small RNA at high cell density. So what Kai did was a northern blot measuring the VQMR small RNA, and he did it using cell-free culture fluids, cell-free supernatants from cholera that had all the autoinducers or that were mutants that were missing one autoinducer, missing the other autoinducer, or missing both. And that way we could test which autoinducer, if any, was playing a role. Okay, so here's the experiment. You're looking at a northern blot. And so what you can see is that cholera, so this is VQMR, the small RNA. And so what you can see is that if we don't add any cell-free culture fluids, there's a little bit of that small RNA there. Then if we take cell-free culture fluids, we grow the cells up, the autoinducers, the entire complement of autoinducers is out in the soups. We take those cell-free fluids. If we squirt them on cholera, VQMR turns on. So that is Quorum Sensing 101. That shows there's an autoinducer in the soup that is turning on this gene. Okay, so now we were in business, and I told you we have these mutants, so we can test which of the autoinducers it was. So Kai used cell-free supernatants from a cholera mutant that didn't make the interspecies molecule, and which we call AI2, and VQMR turned right on. Okay, so it's not that autoinducer. So then he used the opposite mutant, a mutant that didn't make the first autoinducer. This is the one about the genera. He squirted those cell-free soups on, and VQMR turned right on. We're like, okay, it's not that one, but we've seen this before. They act redundantly. So he thought, okay, if I have a mutant that makes none of the autoinducers we know about, that will work. So he took the cell-free culture fluids from a mutant that made no autoinducer that had ever been discovered, squirted them on and the cells, and what you can see is VQMR turned right on. So I've given this away with the title. What we interpreted this result to mean is that there had to be another autoinducer. Cholera made an autoinducer that we had never discovered that's present at high cell density in the cell-free supernatants when we use the mutants we had on hand. Okay, so putting together where we are now, what we would say is at low cell density, when none of the autoinducers are there, cholera is highly infectious. Biofilm genes are on, virulence genes are on, it gets into the host. Then at high cell density, 3 autoinducers, we would guess, accumulate. They bind their receptors, and then together, these 3 systems, in this case through a new small RNA, all of these systems feed information into the cell to turn off biofilm formation, to turn off virulence gene expression, and to turn on the escape program so that cholera can disseminate. And we would have to suspect that there's another autoinducer that works with this VQMA receptor that we had not discovered. So at that point in the game, Kai got a job and left the lab. He's now a professor in Germany. And so he had figured this part out, but then he went to start his independent career. And what was really lucky is that a first-year rotation student named Justin Silpe had been working with Kai on all of the projects that I'm telling you about here. And so Justin decided to join the lab as a graduate student after doing his rotation, and he wanted to take on this autoinducer, this putative new autoinducer, as his project. And so he wanted to figure out what it is. And so he made a really important observation. What Justin noticed is that E. coli and many other species of bacteria seem to make this molecule. And so what I mean by that is we could just collect up cell-free culture fluids from high cell density cells of every kind of cell we have in the lab, squirt them on this strain and ask whether VQMR turns on. And sure enough, it does. So it looked like this putative new autoinducer is broadly made in the bacterial kingdom. Okay, so that's just an observation, but Justin thought he could use that observation to purify the molecule because laboratory E. coli seems to make this molecule. So here's what Justin did. He thought, okay, I can use E. coli sort of as a volunteer. It makes the autoinducer. So E. coli would make this putative autoinducer. And what he could do, since it's laboratory E. coli, is he could clone into E. coli the gene encoding the cholera receptor. Right? So he could put this into an E. coli cloning strain, that a protein production strain, that E. coli would make gobs of this protein, but none of the other cholera quorum sensing stuff, it would be isolated from all other cholera components. So he did that and he put onto this protein a 6-hist tag, so a histidine tag for purification. So the idea is E. coli is going to pump out this VQMA protein. It's making the autoinducer itself. So VQMA will do its job and bind the putative autoinducer. And so Justin could just grow, some liters of this stuff. They're making a lot of the protein. It's bound to the autoinducer. And then he could just pop the E. coli and then put the lysates on nickel beads since there was a hist tag on this protein. So he collects the protein on the nickel beads, hopefully bound to the putative autoinducer. And of course, we don't care about the protein. We already know about the protein. So what Justin did then was he's got the purified protein right on the beads. So he just heated it up to like 60 degrees. And so the protein denatures, it stays stuck to the beads. The autoinducer drops out, and then Justin could just spin the protein in the beads out of the solution, collect up what was left over that would contain the autoinducer, and take these samples, and we could do NMR and mass spec and figure out the structure of the molecule. And he did that. So we asked what it was, and this is the autoinducer. So this molecule, the reason I'm drawing it as a hexagon, it's a 6-membered ring with these 2 nitrogens, and we call it DPO, and that has to do with its chemical name. And so he purified it, and then we figured out a synthetic procedure, and so we made it. And sure enough, if we add that molecule to cholera, it will shut off biofilms, it will shut off virulence, and it will go through the circuit that I told you. So this is the new autoinducer. And what I can also tell you is that Justin figured out the biosynthetic procedure, the cell biosynthetic procedure, and this autoinducer is made from 2 amino acids. It's made from threonine and alanine. So they sort of get pasted together, and half the molecule comes from alanine, half of it comes from threonine. So 2 totally simple amino acids. What I should tell you is this is a brand new molecule to mankind. It's never been seen before. And what is I should have told you is that's true for the other 3 autoinducers. So every time we find an autoinducer, they're really simple molecules, but they're always brand new, they've never been known before. And so I think that just shows that these bacteria are just a treasure trove of metabolic ingenuity, and they make all of these interesting molecules that are waiting for us to discover them and find their functions. So in this case, Justin discovered this molecule, DPO, made from threonine and alanine, and it's this new cholera autoinducer that controls virulence, biofilms, and dissemination. Okay, so now putting this into the circuit, you understand there's this 3rd autoinducer that all the systems work, essentially the same, which is to turn on a set of genes at low cell density and turn them off at high cell density. And so now let me try to put together this part of my talk. All right, so cholera gets into the human intestine. And when it gets there, it's got to decide stay or go, stay or go. And the way cholera makes that decision is through quorum sensing, through several quorum sensing systems. But for today, the new part of that is with this molecule DPO. So DPO accumulates at high cell density, VQMA binds it and launches the escape program, and cholera leaves its current host to infect the next one. You'll remember that I told you that Justin saw that lots of bacteria make DPO. So what he also found is that many bacteria that are part of the microbiome, which is the consortium of bacteria, the 1,000 species that live in the gut, they make DPO. And what I also told you is that DPO is made from threonine and alanine. So it turns out that microbiome bacteria, one of the sources of food that they live on is a protein called mucin. So your intestinal cells are covered with mucin. It keeps them hydrated. It keeps the microbiome away from the surface of your cells, and it feeds the microbiome bacteria. They have mucinases. So they sort of eat mucin for a living. I mean, they're supposed to. So it turns out that every third amino acid in mucin is threonine. So what we would say is that you, the host, the human host, your cells are providing your microbiome the threonine they need to make DPO. They make DPO, and it's known, at least in mouse experiments, that if there's lots of DPO and if the cholera has VQMA, cholera miscounts, right? Because your microbiome is adding DPO, and cholera miscounts and it prematurely leaves the host. That's in a mouse. And so Jeffrey Gordon showed that cholera, if it doesn't have VQMA, it disperses early. We would say that's because VQMA is detecting DPO, right? That your microbiome is working to fend off this invader by making it miscount and disperse early. So we think then that this is sort of a three-way conversation between you, the eukaryotic host, because you supply the substrate, your microbiome cells that make this autoinducer, and the predator, Vibrio cholera. And so you and your microbiome are working hand in hand to try to make cholera disperse early. Okay, so that's in a mouse. It's known that humans get more or less severe cholera disease. A fantasy of ours, and it is fantasy, is that may have to do with the blend of microbiome bacteria there and how much DPO is being produced. That remains to be shown. It certainly seems there's a lot of evidence for that in a mouse, but none in a human, but we think maybe those results will track. Okay, so that's how it's working. That's our new autoinducer. But I told you that Justin was a first-year graduate student. And so after he finished finding the autoinducer molecule, he had to move on to his next project for his thesis. And so, of course, he was captivated by this system because he'd been working on it by then for a couple of years or a year or two, and he wanted to work on it more. And so I told you that Justin made this interesting observation, which is that DPO production is widespread in the bacterial kingdom. What Justin also found out was something different regarding VQMA, the receptor. Vibrios and only Vibrio bacteria have VQMA. So every Vibrio has a VQMA protein, but no bacterium except Vibrios have a VQMA protein. So there was this asymmetry between production of the autoinducer, which seems to be widespread, and our understanding of reception of the autoinducer, which the only receptor we know is restricted to Vibrios. And so Justin was confused by that result. I should say we still don't understand that result, but he was thinking about that, and he was farming around and looking at databases, trying to understand this asymmetric pattern between the autoinducer and the receptor. And so he did find one more VQMA, but it wasn't in a bacterium. He found that there was a VQMA gene on a phage, on a virus. And so this phage is called, I should tell you that phages are viruses that infect bacteria. And so this phage is a Vibrio phage. It lives in Vibrios. It's called VP882. And so this phage came out of a genome sequencing project. Nobody has ever studied it. It's a plasmid-like phage. So it lives as a plasmid. It never integrates into the host chromosome. It just sort of lives like a plasmid in its Vibrio host. And Justin could see that there was a VQMA gene on there. And before that finding, there was no connection, none, between quorum sensing and phages, right? Quorum sensing is supposed to be about bacteria counting, right? And so we got really intrigued by this, like what was this connection between viruses and quorum sensing? So we got our hands on this phage, we brought it into the lab, and Justin wanted to study it. So the phage is about 40 kb long. I just have a little bit of its genome shown here. And what I can tell you is that over where I have on the right, these color-coded genes, there were genes we could understand from classic textbook studies called C1, Q, and then these yellow genes are what are required for lysis. Those kill the cells. And so these we understood from seminal experiments, for example, of temperate phages like lambda. And so let me tell you about these genes first before I get to quorum sensing. So in phages, or the start of the molecular biology field, these classic genes have been studied like from lambda. And the way it works is when a phage gets into its bacterial host, it has to decide stay or go, stay or go. And so that's lysogeny or lysis, lysogeny or lysis. And the way phages or temperate phages figure that out is with these two components. So what happens is if a phage is happily living as a lysogen in a bacterial cell and the host gets into trouble, there's SOS, there's DNA damage, there's stress, the phage says, that's it, I'm out. And what happens is that this repressor called C1 is cleaved. That inactivates it and allows the expression of a factor called Q, and Q turns on the lysis genes. So under stress or DNA damage, this circuit engages, the phage kills the host and moves on to another host. So we could see those genes on our phage, and Justin did the classic experiment, which is put them under stress or DNA damage. And just what I showed you on the last slide happens. C1 gets cleaved, Q is unleashed, and the phage kills the host. Okay, so that is out of a textbook. We didn't need to rediscover that. Many beautiful experiments have been done studying these genes. Our question is, why is there a quorum sensing gene on this phage? And why is our quorum sensing gene, VQMA, on this phage? That's what we wanted to study. Okay, so I told you we had just discovered VQMA, this receptor, right? And so we only knew how to do one thing, which is to add the autoinducer that we had just discovered, DPO, and ask what happens. And so Justin did that. So what he did was he put the phage, remember the phage is a plasmid, we can just transform it into any bacterium. So he transformed this phage into Vibrio cholera. And what you can see, so now cholera has the phage, and this is just a growth curve of cholera. And what you can see is that cholera grows just fine. It grows fine. But remember, I told you we can make DPO, the autoinducer, we can make that synthetically. So if Justin adds DPO to cholera carrying the phage, now look at the white circles, what you can see is that all the cholera die. So what this means is that this phage is eavesdropping. It's listening in to the buildup of the DPO, the host DPO autoinducer, and at high cell density, it kills the host and escapes to the next victim. And so it's a surveilling or an eavesdropping mechanism. And so what you can imagine, so remember a phage, when a phage decides to leave its current host, right? If there's not another host to get into, that phage is a goner. So when's a good time to kill your current host and move on? It's at high cell density. And so what we think then is that this phage is really smart. It's listening into and recognizing when the cells are at high cell density by surveilling the autoinducer. And that's when it kills the host because it optimizes transmission. Okay, so now we think there's this connection between phage quorum sensing and this classic beautiful lysis program that others have discovered. So Justin next wanted to understand, how is this eavesdropping mechanism connected to the genes that we know about that are required for lysis? And so there were some easy guesses. Remember, this is a transcription factor. It's a DNA binding protein. It's an activator. So he thought, okay, if the complex turns on Q, that would cause lysis. So Justin tested that. Completely wrong. We said, okay, we'll just go up a step. If this represses the repressor, C1, that would also drive lysis. Justin tested that. Completely wrong. So now we were out of genes that we understood. So what we had to infer from those findings is there must be some new component that we don't know about that completes the circuit and connects phage quorum sensing to lysis. And so Justin wanted to try to find that component. And so the way he did it was with a genetic screen. Okay, and so here's the logic. So you get, it's all about Q, turning on Q. So and then the cells die, but measuring cell death, that's hard. And so what Justin did was he took the Q promoter from the phage and he put the luciferase genes behind it. So when this promoter engages, the bacteria make light. He put this C1, and the VQMA into recombinant E. coli. And of course, we can add DPO with the pipette. So these cells, these E. coli containing all of this stuff, they're dark because this component is missing and the circuit is not complete. So then what Justin did was he just took the phage genome, chopped it up into bits, and made a little library in a plasmid. And then he shotgun cloned this library into this recombinant E. coli and looked for E. coli that made light. The idea being he would clone the gene that completed the circuit and allowed light to be produced. And so sure enough, that experiment worked. He got his plasmid that had the phage gene that connected the components, and he could sequence it and we could map it back to the phage genome. We have the sequence of the genome. And what he found was he got exactly one gene out of that screen, and it was this one that I have in red. He found there was a gene right next door to the VQMA receptor gene running in the opposite direction. This gene encodes a 78 amino acid protein, so a tiny little protein, and it has no homology to anything in any database this time. And so Justin got to name this gene. He calls it Qtip, for quorum triggered inactivator of C1 protein. So somehow Qtip completes that circuit. And so the next task for Justin was to figure out, what does Qtip do to allow lysis? And he did lots of experiments, and I'm just going to show you the one that gave the answer. Qtip sequesters that C1 repressor. Okay, so let me show you. Here you're just looking under a microscope. This is E. coli, in which Justin has cloned C1, the repressor, and it's got a halo tag on it, so the cells turn green. And so what you can see is that C1 is diffuse in the cytoplasm. If Justin co-expresses Qtip, his new gene, in that E. coli, what you can see is that all of the C1 makes these big oligomers and it drops out of solution at the poles, right? So Qtip sequesters C1 into gunk, and it falls out of solution. And then what you should also be able to know is this is why the protein is called Qtip. These look like little Q-tips to us under the microscope. And so what I can also tell you is that this C1 is active, this C1 is inactive. So sequestration is enough to inactivate the C1 repressor and allow lysis to happen. All right, so putting together what I've told you so far about this phage is that there's two inputs that drive lysis. One is classic, SOS, DNA damage, C1 gets cleaved, Q gets unleashed, and lysis happens. The second input is new. It's phage quorum sensing. The phage is eavesdropping on the bacterial-produced autoinducer DPO. At high cell density, this complex forms. It sits at the promoter of Qtip. Qtip gets made, and Qtip sequesters C1. It drags it out of solution and inactivates it. Then Q is unleashed again, and lysis happens. So the same thing happens. The cells get killed. But the fate of C1 is different. In one case, it's cleaved. In one case, it's not. And so this is really new. To our knowledge, there's never been any input other than SOS or DNA damage controlling phage lysis programs. We would like to say that quorum sensing can do that as well, at least in this phage. Okay, so there's two inputs. But also, you'll remember from the beginning of my talk, there's two quorum sensing systems running here. Remember, cholera is doing quorum sensing as well. Cholera has its own VQMA, it binds DPO, and then at high cell density, it turns on the Chi's small RNA, and that small RNA controls 600 genes, right? So that's host quorum sensing. What we showed is that this phage quorum sensing circuit also controls the host biology. We don't understand what good it does the phage to control all 600 of these genes. It's about to execute cholera, but in the meantime, it messes with all of this host biology. We don't understand why that's beneficial to the phage. We just know that it happens, and we're trying to figure that out as well. Okay, so two mechanisms for lysis, two quorum sensing systems running in parallel, both of which are responsive to the same autoinducer. All right, so once Justin got this far, we were intrigued by this phage, but we started to worry that this is just some one-off, that this one phage, has our receptor, and maybe it was some accident of evolution. And so we wanted to see if that was the case. And so now that Justin had this module with 2 genes, he could do better database analysis. So he went back to the viral database looking, and what he found was, sure enough, this is not a one-off. There are all kinds of plasmid-like phages that are totally understudied, like they just haven't been studied, all of which have a transcription factor, so the purple gene, and then right next door, they have a tiny little gene, that I have in red. And so these transcription factors are all DNA binding proteins, and they all bind a ligand. We don't know what these other ligands are. These red proteins, they have nothing in common. Remember, this top one is Qtip. They do not have amino acids or anything in common, but they are functionally equivalent. So what we did was we cloned all of these red genes out, and every one of these red genes will sequester every one of these C1s. Okay, so it works the way I'm telling you in all of these phages. But the problem is we don't have these other phages. These are just, data A's, G's, T's, and C's out of database analysis, that Justin put together, right? So we don't know the hosts. We don't know what inputs they're listening to. But what we'd like to say is that this idea of interpreting host sensory information is going to be common. So in the phage we studied, that phage listens in to the autoinducer DPO, and with this Qtip anti-repressor mechanism, it drives lysis at high cell density. In all these other phages, we don't know what input, the receptor or the transcription factor monitors, but probably some appropriate host biology, and then by turning on its cognate anti-repressor, lysis happens, right? So what we think then is that these phages are communicating with their hosts, and they're taking in presumably all kinds of sensory information that's important to the biology of the hosts in which they live. And we would love to know who those hosts are, and we would love to know what these signals are, but we don't. Okay, so for the final thing that I'll tell you is that when Justin found that, he thought, what these phages are doing is they're re-engineering this circuit to be responsive to different signals. And he thought, if the phages can do that, well, I can do that. And so what we wondered is if we could take this basic foundational knowledge and do something practical, which is to make a little application. And so what Justin did was he made a whole set of toy phage therapies that allow us to kill pathogens, any pathogen we think, on demand. Okay, I'm just going to show you one, and you can use your imagination. We hope that this might be useful to people who make applications. All right, so what you get is that we have this phage, and I've told you that it's a linear phage, it's a plasmid, so we can transform this into any bacterium as far as we can tell. It's not restricted to Vibrios, even though its real host is Vibrio. So we can put it into any bacteria. And what you get about the phage is whenever Q turns on, the cell is going to die. It promotes the lysis program. So what Justin did was he took his phage, the whole phage, and then he just put different promoters in front of the Q gene on the phage that he could control. Okay, so he made 10 or a dozen of these. I'll show you one. He put a promoter called INV-F. This is for invasion, and this promoter comes from Salmonella typhimurium, right? So that has nothing to do with Vibrios. So in the Salmonella, when Salmonella is under virulence conditions, it gets into the intestine and it's going to invade human cells. Under virulence conditions, a Salmonella transcription factor called HIL-A binds at the INV-F promoter and turns on the invasion genes. All right, so now Justin has put the INV-F promoter on the phage. So what we did is we popped that recombinant phage into Salmonella. We put Salmonella under virulence conditions, and HIL-A did its job. It bound the INV-F promoter. The problem was the INV-F promoter is on the Q gene, and so then what happens is that all the Salmonella committed suicide, right? So HIL-A turns on INV-F in the genome, but it turns on INV-F on the plasmid, on the phage, and all the Salmonella lyse. Okay, so you get it. If you have a promoter that you can control with, I don't know, a Arabinose, IPTG, any promoter that you can control, all you have to do is put it in front of this Q gene, and you can target the bacterium of choice on demand by adding the stimulus. So these are not real phage therapies, but we hope that colleagues who actually work on things like that might find it useful to have a phage that kills on demand and has incredibly broad host range. All right, so getting back to the basic stuff, let me finish today's talk and put together both halves of the seminar. So what you get is that cholera, a pathogen, a predator, gets into the human intestine and it has to decide stay or go, stay or go. And it decides that with quorum sensing, including with this new autoinducer DPO. At high cell density, DPO accumulates, cholera recognizes that, it says it's time to leave and go to the next victim, and so it escapes. Your microbiome and you are in part of that conversation because buckets of DPO get made in the intestine, and that we think is your microbiome and you trying to fend off this predator to make it escape early. We presume that DPO is used and sensed for other things by the microbiome. We don't think it got made to, fight off cholera. That's a benefit, but we don't know what the microbiome used DPO for at this point. But now we have to add another kind of organism, the phages, to this mix. So the parasite of the parasite is also using DPO to decide stay or go, stay or go. And so it's monitoring DPO to decide when to stay in its current cholera or when to kill that cholera and go to the next host, right? So all of these organisms are trying to optimize off of this little molecule that I told you is just made from threonine and alanine. And what I have to finish by saying is that now we no longer understand how anybody counts precisely. Since all these organisms are making and contributing DPO and trying to use it, we don't understand how they get accurate information since more than one player is making DPO. And so the task for us right now is to sort of get out of the flask with single species, and we're trying to do experiments that look a lot more like this, where we have multiple species of bacteria, we have the phage present, we have cholera, we have human cells. We're trying to understand how does quorum sensing really get deployed and really work in scenarios that are much more authentic to real life than what we have done in the past. And so that's sort of the challenge for us. It's a fun challenge for us to take the parts and the mutants and the things we know and really try to up the complexity in space and in time, right? And in sort of authentic situations so that we can really learn how quorum sensing gets used in this kind of a scenario and lots of others. And so that's what the lab is working on now. And the jury is out, but hopefully in a few years, I'll be able to tell you more about quorum sensing in real live situations. And let me finish by telling you about today's two heroes. So this is Kai Pappenfort. He's a professor in Jena, Germany. He discovered the VQMR and the VQMA and worked on the autoinducer. And then this is Justin. He is the one that discovered DPO, and he did all of the phage work that I told you about. He's now a postdoc. And one more time, maybe I'll stop sharing and you can perhaps see me. One more time, let me just thank the Microbiology Society. It is such a pleasure to be able to give this talk, and of course, a real honor on behalf of my group to win this prize. We are absolutely delighted and honored, and it's really, it's just terrific. And so I thank you, and I will be here in live to take questions. Thanks very much.